Yeah, so we pretty quickly going to jump into an example, so I just want to check that everybody has the files. Okay. Yeah, so I'm going to do one example now, and this example was kind of supposed to be, it's kind of associated with the work that we did yesterday, and then we'll head into new work from there. And it's just a quick discussion on object recognition and tracking. There's a lot more that can be said about this, and there's no scope really in this course for that. But just to mention two applications for it. So often you have particles that you want to track. Sometimes these particles can be really, really small and you want to see how they move throughout the cell. Other times they're nuclei that you want to maybe track in the scene. But one way that you can do this tracking is using something like TrackMate. It's not listed on the slide, but in Fiji there's this plugin called TrackMate which allows you to set up some conditions and then it tracks things across time. You can think of this kind of tracking similar to what you would have with a CCTV camera, where there's like a bounding box just put around the person. It's like, okay, there's a person, they're moving around. So it's not necessarily very precise. It's not supposed to be the size of the bounding box or the boundaries has to be perfect or something. It's just, you just want to know the position of something. So that's tracking over time. And particles and nuclei, they are usually quite easy to track. Something like mitochondria, they're quite a lot harder to track. A little bit later this morning, I hope to get to just mention a method that can track mitochondria, how they move around in the cell over time. But it's some things are harder to track than others. I had someone reach out to me. I think it's called Z-lines when like the muscle forms or something. Not a biologist. I don't know all the right words. But he was showing me all of these little particles, and then they're forming these lines. And it's like, hey, we would like to track which particle ends up where. And it's like literally a single pixel that needs to be tracked across time. And it's just like this, there are limitations. If your resolving power, if your resolving power is not enough, usually the tracking is not robust. So at least have some kind of a structure that you can identify, and then you can then usually track it over time, not with too much effort, depending on what it is. Then the other thing is with object recognition is we just want to know how many of something there are. We don't necessarily want to have all of the details of the size and the shape descriptors and everything that we've looked at. We just want to know how many there are. I think my friend at the back was talking about just cell counting. Just needs to know how many. What is the ratio of one type over another type? And these could be foci or centromeres or vesicles or whatever you want to find and to track them. And then once you've found them, you can decide what you want to do. Do you want to then do this region growing thing where you then expand your region to select the entire structure for segmentation purposes, or however you want to approach it. But yeah, we're just talking about counting at the moment. So a quick example with this, and you can open up, it's the first folder on day 2. It's the centromeres. Okay, so we want to know, for example, just how many centromeres there are. Or just know the position, relative densities and stuff like that. Now, before we do anything, I think it's always a good idea just to make sure that we can actually see what we're looking at. So just opening this window again, I usually just press Control-Shift-C, but it is under Image, Adjust, and then Brightness Contrast. And then you get this little window. So as we did yesterday, you can either drag the slider, or you could just simply click Auto, and then it should automatically brighten them up for you. Okay, now I must first think about a little bit what am I seeing here. I'm seeing little dots. Some of them are nicely separated. Some of them are connected to other dots in a denser region. Now, if I see something like this, where they're obviously connected, I wouldn't be able to uniquely identify them. So my count will be off in that sense. So just first of all, realizing my image has a limitation, I will not get an exact count, so it's always going to be an estimate, but I can just think about, okay, well, what might that estimate mean? Now the next thing is, what is actually going on with my intensities? So where I have these foci, these centromeres, I believe it is, you can see they have a dim area around it, and it grows brighter, and then dim again, and then brighter. So if I was to find a way to find these peaks, essentially, then I can possibly count them. Now there's a method built into Fiji. It's one of the core methods, which is called Find Maxima 2D. And the way that it kind of works, or the principle under which it works, is you define something called a prominence. And the prominence is basically how, for the closest hill, how far is the difference between it. So let me get this laser pointer. So the prominence from this to that one is this height here. So you always talk about from one peak to another peak, what is the prominence difference? And the prominence is basically how much does it dip down? What is the drop that it takes before it goes up again? I think this is kind of a good example here, because if you were to set okay, maybe just explain what we're going to do now. We're going to choose a prominence value, and then from this prominence value, it's going to say, okay, has it dipped far enough to consider this a separate peak? Okay, that's where we're heading. So you can now see if I've got something like this, then it's very possible that it's not dipping far enough for peak B and C to be considered separately. So if I said, no, I need a higher prominence before I would consider this a separate peak, then it will just simply clump these together as one peak. Where if I make my prominence a little bit smaller, then I can start to identify, oh, okay, peak B is separate from peak C. Because it has to do everything with what is the relative drop from one peak to another peak. Let me just check. Yeah, so let's just quickly find the prominence in my image here. So it doesn't really matter, I'm just going to, where I'm at, just look at that. So what you first want to do is you want to just see what does it look like? What do these dips look like? How much is that dip in a general sense? Maybe I'm just going to try to find a slightly different cell. Oh, come on. No, I'm not in the right focus. I'm trying to find a good representative one, but also where there's a little bit, this one is probably okay. If I draw out a line now through this, let's say I just draw it through those two, just as an example. I just want to get a feel for it. Then I can do the same thing we did yesterday, where I say analyze and then plot profile. And then I can see what the profile looks like. So now I can see, okay, the prominence, in other words, the intensity drop from one peak to the other peak, it goes down with about from, let's say, about 180 to about 140. So it drops with about 40. But that's a pretty clear drop. Let's just see if I was to look at some other area. Okay, now shift this around. If I get both on the same screen. If I just move this line around, let's say, for example, that one, it's a little bit less clear there, what is the one to the other. So it goes down from about 175 to about 145, so that's about 30. But I might even want something that's a little bit more subtle, like I want these to be counted as two separate ones. And that drops from about 165 to about 145, so it's about 20. And I can now go on with this process and then decide, okay, what is a good prominence threshold to actually consider something one peak separate from the other peak? You know, once I've decided what that value is, I'm just going to choose 15. I think 15 would be a pretty safe value. Then I can click here on process, find maxima. And then I say what this prominence should be. Now I'm just saying I feel like it should be 15. And then let's just preview what it actually does. Okay, no, I don't know if you can see this very clearly. Maybe I'm just going to zoom out. It might be a bit more obvious. Process, find maxima, preview. Yeah, there you can see it a bit more clearly. So these are all the peaks that it has found now. And it tells me here it's found 1,707 maxima. Now you can see that some of them are kind of not really what I want, like these dots there. I don't really want, that's basically noise. So maybe I need to pre-process the image a little bit, clean it up a little bit before I actually do this entire thing. You can obviously always do that. I'm just showing now the raw approach. So what would happen if I change this prominence? So let me just quickly think about it. If it was to make this prominence less, like let's say to 10, would you expect this number of maximum to increase or decrease? Yeah, so let's see, it's going to increase because now the prominence is lower for it to consider it a peak. It's not forcing it to go as far down for one peak to another peak. If I make it more, let's say 20, now there are fewer peaks because it has to go further down before it actually considers two peaks separate from each other. And you can obviously now again play around with this and look at actually what's happening to your image until you feel like, okay, this is a pretty fair representation. You see, just changing it from 15 to 30, it has changed from 1,700 to about 1,000. But let's just leave it at 15 for now. Okay, now I've got all of these selections. These are point selections, so I can do the same things I did yesterday. I can go to Tools, ROI Manager, and then add them. Now, interestingly enough, it just adds one item, but this one item actually corresponds to everything. So if I click Measure, it might take a bit of time. Yeah, there you go. there you can see it's found all 1,707 of them. It has listed them individually in my measurements. Now you'll notice that the minimum and maximum are obviously the same as the mean because there is only one dot. It's only sampling one dot. It's not selecting the entire structure, but it's you can still do interesting things like because these should be the peaks. So what are the average peak intensity or things like that. And then you can use the XY coordinates. These are the XY coordinates of where they are found. You can then talk about densities, like how close are they together in different vicinities or something like that. Just one more thing, maybe to show is if I let's just clear the selection, edit selection, select none. Okay. If I was to only want to know what is the number of peaks in a specific structure, so let's just take one somewhat at random. Just going to take this one here. I could first go and select this structure. Just it doesn't matter in this case to have a very precise selection. Okay, and then I can. After it's now selected, then I can say find maxima. And then you'll see that it's maybe not that clear, but let me just generate it. You'll see that it's only generated the maxima in that structure that I selected. So what selections usually do is it limits whatever operation you perform to that thing. The lookup table is obviously the exception to that. But if I only want to have the items or the peaks detected in a certain region, I can first make a region of interest selection, and then whatever analysis I do will only be for that region of interest selection. So I can do the same. That's now my old one. Let's just start over again. Shift, come on. Okay, so make a selection, and then find maxima. Right, and then I can go, first add it and then measure it. And there are the 50 that was found in there, 55. Wow, it doesn't seem like that many, but apparently it is. Yeah, so you get the feeling if you just want to get object counting, this is a kind of a useful way, because usually if you've got nuclei spread out, they would follow the same idea. There would be some intensity dip before it reaches the next nuclei, so you can just set that up. Okay, so now we're getting into the more, I suppose, meaty part of today's talk. This is all the things I showed. Great, so we're talking now about deconvolution and what is necessary when we acquire cells. Now, just to explain the general approach of microscopy, so you obviously have your cell or your sample, whatever you're trying to image, then you've got your sensor at the top, and the light needs to get from your sample all the way to the sensor. In the process, there's a lot of lenses that it goes through. Light refracts, different wavelengths of light reflect differently through a lens. So there's that component. There's also some distortions that a lens can introduce. So no lens is perfect, no refraction is exactly how you would expect it. So a lot of random noise, it's not really noise as much, but it's a blurring or a non-specificness that enters into your image due to the effect of the lenses, the refraction through the lenses, and it not actually ending up very clean on your sensor the way it was actually imaged. Here's just a quick example of what typically would happen is you have this perfect sample under the microscope, it's perfectly stained, but then when you image it, due to, like I was just explaining, the lenses, the refraction, and all of these things, as well as just some additional noise from quantum effects and strut noise and all of these other things, you end up getting this somewhat blurry image. And this is now what you have to analyze. So that's obviously not very desirable or ideal, but the great thing is we can model that amount of distortion that the microscope introduces. It's not like shot noise, for example, is difficult to model because it's somewhat random, but the distortion that the microscope brings into your sample is fairly consistent. It could be slight variations depending on the oil, on the objective, and all of these other factors and temperatures and stuff can all affect it. But in a general sense, it's pretty consistent. So if we were to model what this distortion is that the microscope introduces, we would be able to mathematically undistort it because we know what function was added, we know how to unadd that function. And this is the rough picture to say. We've got our object, that's the thing we're trying to image. And then there's this thing what we're going to call a point spread function. I'll explain just now what that is. But basically when you image, these two things become convolved together. What is a convolution operation? Think about that kernel that we shift around on the image. So it's a certain operation which is performed across your entire image, and if it's in 3D, it's also in 3 dimensions that it's doing this, and then it leads to this distorted image. So after the convolution, this thing with that thing, you see that all the structures, because this is a long elongated thing in this example, everything becomes elongated, and everything becomes slightly blurred. And then also, as I was saying, you have all of these additional effects of noise and things that just temperature differences can cause quite a lot of additional background noise in the image. Okay, so this is now the image we get, and we want to get as close as possible back to that image there. So let me just get into this point spread function a bit more. What a point spread function is trying to describe is if I have a single point, like just point intensity underneath my microscope, and I was to just image that point. I know it's a single dot, it's supposed to be a dot. You try to make the point usually as large or just slightly smaller than the iris resolution that you can capture. So it's, you know, it's going to be a single pixel, that's the point. And then the single pixel then obviously becomes distorted and enlarged and everything, and then you say, okay, well great, there's this entire structure that I'm now seeing. And then that is that structure is the point spread function, which is like, how is this point becoming a larger thing through my microscope as it's being imaged. So if we then know what that point spread function is, since we did a convolution to generate our blurry image, we could just do a deconvolution, which is the inverse mathematical operation, to recover our image as close as possible to the original. Now, since there is noise that we don't know what it is, we will never recover it perfectly. We will always have to kind of accept a somewhat suboptimal reconstruction. And there are other things such as the point spread function is usually either not consistent between image runs, or it's estimated. We don't actually know what the point spread function is, and I'll talk about point spread function estimation just now. So a lot of things in the deconvolution is not perfect, but it's a whole lot better than what we had if we didn't do deconvolution. Now, as you can see, since this convolution with this point spread function of our image happens pretty much immediately in our entire image acquisition pipeline, before you do any further pre-processing or analysis or anything, the very first thing you need to do is undo this convolution by doing deconvolution. Now, I was chatting to Lisa yesterday. Most microscopy software can have it included, but you have to pay extra for it. So the people that manufacture the microscope obviously know the microscope very well, so they've set up all of their software to have as close as possible the best point spread function set up for that microscope and the best deconvolution algorithm for that microscope. And then you capture the images, they are automatically deconvolved, and then you have your clean image that you get out of the microscope. That is not always the case. I know it's not the case here, just due to historical decisions and understanding of things. But the reality is that you need to make sure that your images are deconvolved before you analyze them, because trying to get sense out of this is rubbish. Getting sense out of that, you actually get a little bit more concrete information. Here's just another example to hopefully drive the point home. It's always more clear on my screen than it is on yours, but if I just compare that region there, for example, it's just a blurred mess. I have no idea what's going on there. But after this deconvolution, I can actually see I can kind of see the strands in my cell. I can see the structure. I can also see that these yellowish red areas is actually small dots of interaction, where that information is entirely lost in the blurred image that you get directly out of the microscope. So very important, always deconvolve your samples before you actually analyze them. I'm not going to go through this. I basically said it. It's just the idea of your image is basically a function. It gets degraded. You have some additional noise, and then you just need to try to restore it, which is the inverse operation, to get an estimate. This is, you see that little hat there. The emphasis is this is an estimate of what I had initially. I can't ever get back to perfectly what I had, but it's an estimate. Right, now what does a point spread function potentially look like? Well, it varies entirely on your acquisition parameters. So there are certain software things, I've put some links here as well, where you can fill in your acquisition parameters, what type of microscope did you use, what was your numerical aperture, this is usually about 1.4, what was your excitation wavelength, emission wavelength. I haven't really discussed this, but it's maybe just something to, I could probably find a picture now, but the rough principle here is that you put a fluorophore or something in your cell, and it binds to the structure that you're interested in. Now you need to shine a certain color laser on it. It's designed to work with a certain color laser, that if you shine that light laser on it, it's going to be excited, and then it's going to a higher energy state, and then it's going to fall back to a lower energy state. And that falling back releases some photons. And then as these photons are released, we perceive them as a certain wavelength of light. But usually, the energy that you get out of the system is less than the energy that you put in the system. So you would excite with a higher frequency, and you would get a lower frequency light out. Or you would excite with a low wavelength and then get a higher wavelength out. So frequency, wavelength, they're inverse. But yeah, this is what this means with what laser frequency or wavelength that you excite your structure, and then what did you actually get out. What you got out is a whole lot more important than what you actually put in, because what you get out is what is going to go through the lens all the way to your sensor. So yeah.
The fact that you actually get a range of emission, does that affect the...
Yes, it does. So ultimately, when you set up a point spread function, artificially you set up for a specific wavelength. So if there's a range of emission, you have to say, okay, well, what is either where is the peak, because usually they show you there's a peak in this range, and then you try to use that peak. Otherwise, if you don't know what the peak is, then you just say, okay, well, let's just split the difference. It's in this range. Let's just choose the middle value. But yeah, you need to set up the point spread function specifically for the emission wavelength. Then I'll demonstrate that now with an example. And yeah, then the refractive index, that is obviously one of the main sources of this distortion, and that's dependent on is it immersed in oil or whatever other media you might use. And then the specimen itself could have the refractive index, and the pinhole radius, all of these things affected. Now, for the most part, we don't need necessarily all of this information if you want to estimate the point spread function, but if we have everything, our point spread function estimation would become increasingly accurate. And this is what a point spread function might look like. The different colors, they're not actual colors, it's a color map, so it's just intensities that's now just with the lookup table represented as the colors. And you can see that it's got both from the top view, it's both got this radial pattern, and from the XZ view, like this one from the side, it's also got some pattern. So it's not just a two-dimensional thing, it's a three-dimensional distortion that you have. So what that means is, if I'm going to convolve dimensional structure over my Z stack, for example, I will be including some of the information of lower down layers into the layer that I'm currently looking at. And the slice that I'm currently looking at, some of the slices below and above, that information or intensities will be poured into this central layer. And what deconvolution is trying to do is trying to throw that out again so that I only have the information for the slice that I'm actually looking at. So how do we get the point spread function? So the one is theoretical through math. I'll get a little bit more into that just now. The other one is where we actually put these point sources in our microscope and then we image them. Like I was saying, sub-resolution spherical fluorescent beads. So the idea here is that you put a bead in which is smaller than the resolution that the microscope can capture, so that it's supposed to show up as a single pixel. And if it's a single pixel, you've got no distortion. It will never be, because we always sit with this. But this is what you might get out. You get an image that looks like this, and you can think of each of these dots basically as a representation of what my point spread function is for this microscope. And this is now obviously in Z, so it's three-dimensional. It's just not showing it here in 2D. There are some approaches you can take where you say, okay, well, these two, they're somewhat overlapping the information, so I don't know, actually, I can't separate them cleanly, so let's just say that they're not so good, I don't want to reduce them. So I try to find the best three most representative, in this case now, examples, and then I can average their values to get my point spread function out. That is the one that I then say, for this microscope, for this setup, this is the point spread function I should be using to get the best deconvolved result. Now, for a few reasons, this is not always necessarily possible. It's either expensive or it's time consuming, or there's just too much variation. If you were to do it in the summer, or you do it in the winter, or you do it slightly, I don't know, the different microscopist is doing the actual work, this point spread function might vary a little bit. So just extracting it once is probably better than just calculating it theoretically, but it's not going to be consistent. And always just be aware of all of those nuances, that it's an estimate at the end of the day. Yeah, so it's also interesting, if you do this, we extract it experimentally, to always compare with what you calculate theoretically, because what might happen is, if you if you were to apply a theoretical thing to this, you might find that one dot is shifted, well, in this case, the dots are not shifted anymore, but by having a measured PSF, there might be some translational shifts that actually happens in your structure. So it's not just unblurring, it's possibly even a slight translation thing that you could apply there. And if you had the measured one, you could possibly, in this case, as you can see, distinguish between two point structures, which with the theoretical one they were indistinguishable. So yeah, just it's kind of interesting, good to compare, just to maybe see whether your measured one is actually as accurate as it should be, and also if you don't have a lot of trust in your measured one, then you say, well, is my theoretical one sufficient? It's a good comparison to make.
For your measured point-spread function, would you have to do a fluorescent bead for each channel, or can you do a...
Yeah, so this is also one of the reasons why it's not often done, is you have to do it for every excitation wavelength, emission wavelength, sorry. So if you have a different stain, that's going to have different emission properties, like different range of spectra, then it's going to have a different point spread function. So every time you do a new experiment, you basically need to generate new point spread functions. So for each channel. So it's not very practical, but if you want to have absolute accuracy, this is the way. Yeah.
We're using the bead slides, they could have very different intensity than the actual sample. Does that affect the accuracy as well?
Not so much the relative intensity, no. It's more just the distortion or the blurring that you actually want to model. Yeah.
But you should use the right objective for the right oil, with the right emission.
Yes, exact same conditions as the actual sample you're going to image. Okay, now doing this, once you have your point spread function, different ways however you might get it, then you actually need to do the operation of deconvolving your sample. Now, it's a very time-consuming process, especially the larger the samples get. You can imagine if it's a small sample, I need to shift this thing back and forth over the image only a few times, then I've covered the full image. If my Z-stack gets deeper or my sample gets high resolution, it takes substantially longer, and it usually doesn't scale very linearly because obviously I've got an X and a Y axis, so it scales quadratically, I think. Could be wrong on that, but it's not favorable. It's going to be quite slow. So you've got a lot of options as to the quality that you want out and the time that you're willing to wait for it. Generally speaking, just wait longer, get better data, get more statistically accurate results. But to give you a spectrum of what's available, you have these things called inverse filters, where you take this point spread function and then just try to apply it directly to your image. You basically just directly do the deconvolution mathematical operation onto your image. And there are different approaches to this. You can now look them up if you want to. Just a short list of them. There are more, but this is just some of the more prominent ones. And then there's these iterative methods, where what they're doing is they do a little bit of deconvolution, then they compare how does the image, what is the result? Okay, well, let's do a little bit more. How should I adjust things? So it's an iterative thing where bit by bit cleans up your image. And there's a balance in this usually as well, because there are some parameters that you should tune, and we'll see that a little bit later. So you can regularize it to say, don't change my original data too much, just try to still retain some of the structure a bit more, so that you have this regularization parameter. Or you can say, how many iterations should it run? If I can't wait very long, I might only run it for 5 iterations, then the image might not be as clean as it can be. But on the other hand, if I run it for 50 iterations, it could be that I'm now starting to destroy actual structure, because I'm now telling the thing it must keep on removing stuff even though there's maybe nothing more to remove. So there is always a balance in the choice of parameters that you choose to get the best result. But in a nutshell version, if you use these inverse methods, they're very fast. They don't require as much computational power on your computer, but they usually leave quite a lot of artifacts because it's not considering what happens to the sample progressively. Where the iterative approach is much cleaner, much more defined structures, but they are slow and more computationally intensive. Just some examples of what some of these methods might produce. This is not my data, but I want to share some of the projects that we've been working on in terms of this as well. So you get your raw data out of the microscope, very blurry, and then these are some of the methods that you could apply. Now you can see for all three of them, they produce slightly different results. So let's just take that structure at the top there. This is still somewhat of a blurred mess. For these two, especially this Richardson-Lucy, it's cleaned it up quite a bit. You can start to see very sharp edges in your structures. some other things like that one now, for example, here it is a very bright spot where with these two iterative approaches, it's a fairly dim area. So just be aware that there is no best result. There is a judgment call that you have to make to say, what am I kind of visually expecting to see, and after deconvolution, did it clean it up in a way that I think is appropriate, that I deem the sharp edges, but it didn't destroy the structures. It's highlighting the things that it should be. It's like pushing away the noise. It's a very subjective judgment call, in all honesty, to decide what parameters to use, to decide what method to use, etc. And I just want to show you, like I was saying, some of our experiments here. So we started this project where we tried to train a neural network that can do the deconvolution for you, because it's so computationally expensive and it's extremely slow. And if you have a lot of data that you need to deconvolve, you literally put it on and you sit for days. If you just wait, the thing is just deconvolving in the background. So a neural network is incredibly fast. It's like 1 or 2 seconds done, the deconvolution is done for one image. So you definitely want to leverage that speed. The hope of a neural network is, neural networks are known as universal function approximators. So in principle, if there is a function from the input to the output, it can approximate what that function is. It can learn what that function is, and then it's a very quick input-output. It just does the math and gets the answer. But the big question here now is, can we actually learn that mapping? I realize I'm not in the right paper to describe that, but this paper is, this thesis scripty will still be good for another discussion. But that's the context of the project. We wanted to train a neural network. I'll show you some of the results a little bit later. But in order to train this neural network, we first needed to create the ground truth, which is the input-output pairs. We're putting in our images, and we're getting out perfectly deconvolved images. But we need to decide what deconvolution method are we going to use, what point-spread function generation method are we going to use. There are so many options available. So here is just some of the results. You'll see some of these words come up a little bit later as well. So you can kind of ignore the right-hand side image. So the top, this is now just the raw intensity image. This is where the intensities are just extracted for that mitochondria channel, and we want to look at the structures as they come. So this is the raw image. This is what we got from the microscope. And then we first checked, okay, well, if we apply a specific deconvolution method, what point spread function generation thing should be used? Because we are going to artificially estimate this point spread function. We don't know what is the best one for our microscope. So you just kind of try all of them. And then you see for this point spread function generation, it kind of like destroyed my image, so not great. This one's reasonable. It's cleaner, it's good. And then this one, I would say, is probably the cleanest. The edges are the sharpest, and I can make out what I want, and it doesn't feel like it's destroyed my structures. So in our experiment, we found that the boar and wolf, boar and wolf one is the point spread function model that we will be using. And you'll see these words come up just now. And then, okay, now that we know what point spread function to use, then the question is what actual deconvolution method should be used. These inverse filtering ones, or the iterative ones, or things. And here are some of these results. So if we take again the raw image, just keep that picture in your mind. We obviously want to make it better, not worse. So using a regularized inverse filtering, the image looks substantially worse. There are now these fringe elements happening around it. This one made it blurry. It's maybe, I don't know, it's not really better. This one, this Richardson-Lucy one, is pretty clean. Like if you just try to compare these two, it's lowered the intensity, but that's easily fixable. But it's made this thing substantially more clean. And then you can now compare the bottom one, which is Richardson-Lucy with total variation. Now it's very subjective, but after more investigation, we found that this Richardson-Lucy with total variation produced, in our case, the cleanest result. So this is, if you want to do this rigorously, this is how you need to do it. You need to say, I don't know what is going to be the best for my data, for my images, so let's try all of the options, variations, and play around with them, and then we get a feel for what they do, and then you say, okay, great, now going forward, this is what I'll be using for this type of data. So my suggestion to you is use Richardson-Lucy with variation regularization, but please don't just take my word for it, also experiment for yourself. Okay, so back to the slides. Just some, I kind of just mentioned this, but some other ways you can do this is a blind deconvolution where you don't actually have a point spread function. You kind of estimate the point spread function directly from the input image, generally this doesn't produce good results, not surprisingly, but it is a method that's available. And then, as I was just mentioning, these artificial intelligence methods, and I think, yeah, here's an example of after we trained this neural network, it's a image-to-image translation method that we used. And if you consider now at the top are the raw images, you can see that these are 3 different structures, 3 different wavelengths of light. This is emission wavelengths. And then after putting it through our neural network, which has now been trained, we get these results here. So each of these were generated basically within 1 second, so very, very quick. The big question now is, am I going to trust it? And am I going to trust it on different data? That's another question. So what we did do is we would train it on one wavelength of light and see how it generalizes to other wavelengths of light. And we found that it works generally well. But if you train it on all the wavelengths on the 3 different wavelengths of light that we wanted to test on, then it definitely performs better. So a rule of thumb, if you want to go neural network approach, is what you train it on is where it performs well. What you don't train it on, it's a little bit of a guess whether it will work or not. Now, just getting back to my earlier point, am I going to trust this or not? So if I just look at this last one, for example, I need to go now and look with my eye. This is the raw image. I know this is distorted and blurred and everything, but is this a reasonable representation of that, or did it throw away some data? I need to make that judgment call. You need to, in any case, make that judgment call with deconvolution. So it is largely based on your judgment call as to whether this is overly destroying the data or whether this is cleaning up the data. And if you feel like, okay, this is actually a useful sample representative of what is actually going on under the microscope, then you analyze that and you compare results. You just need to be consistent. Everything is apples to apples comparisons. But if you feel like, okay, no, this neural network maybe didn't learn quite well enough what I needed it to learn, maybe I need to just still go back and deconvolve the samples manually, then I have a bit more of a mathematical process. It's maybe even more trusted by other academics because you can tell them exactly what you did. It's not this black box thing which they don't fully understand. So it's a trade-off. You get speed, you get pretty good results, but you lack maybe the mathematical rigor to explain how these results were generated. And maybe as we're talking about this, it is also good to just recognize that deconvolution does change the intensities. So usually what we would do is, because it changes it quite dramatically. So you might start, let's say you start with a 12-bit image, the intensities are between 0 and 2000, for example. Then after deconvolution, the intensities could vary between 1 and 10,000 sometimes. It's a very crude operation for intensity values, but the relative intensities are reliable. It's just the actual intensity becomes quite different. So it's usually a good idea to just normalize all the images to the same range at the end of the day, do some contrast stretching or something to say, okay, consistently, I have shifted these intensities now to be within a range that I know and understand. But it becomes a bit of a challenge because you're always, when you're toying with intensities, you are possibly changing your data. So deconvolution is a messy operation. It's not as clean as you might hope it to be. And there are some future, subsequent considerations of how do I then redistribute my intensities to be sensible for analysis. But just in all fronts, intensities are in any case a very relative thing. It depends on the time of, like how long the shutter is open. It depends on how many times did you scan over your sample or whatever. There are so many things, variables going into the value of intensity, that the value of intensity is not meaningful in and of itself. It's the relative intensities that are of value. And deconvolution doesn't change the relative intensities in an incorrect way, generally speaking. So yeah, maybe that gets close to an answer. All right, so then, I think this is just another example of the same thing. And one of the main reasons why we want to do deconvolution is if I want to do, for example, a structure count, it's going to be difficult to count the number of structures I see here because they're all blurred together, where if it's separate, if it's now cleaned up, I can suddenly see, oh, this is where the structures actually are, and then analysis is more accurate. So I think I've made that point, but that's the reason why we deconvolve, is so that we can make sensible analyses from that. Okay, so now we're going to actually play around with this and see how it works. I did, at the beginning, before the course, I did mention that there are these 2 plugins that you need to install. I think I did put them in the folder here. Where is it now? I think it should be in day 2, deconvolution, plugins. So you should see them there. So you can download them from the links that was emailed to you, but there they are already available for you. Now, I did briefly show how to install plugins yesterday. The point is that these jar files just need to end up in the version of Fiji that you're using in that software's plugins folder. Now, if you don't know where you installed it or you don't want to copy and paste things over, Fiji does allow you to fairly easily just click on plugins and install. And then you just need to find them. Deconvolution, plugins, so that one. And then it basically just says, okay, save it into the plugins folder, save. Okay, I already have it installed, but yeah, that's how you install the plugins. So you just need to do that for both. Just showing you again, it's plugins, install, and then you just find the folder of it, and then you select it, say open, and then save. So just, I'm going to give you just a moment to do all of that. And then once you've done everything, you just need to close Fiji and reopen it. Otherwise, it's not going to reflect. Yeah.
So, I've got everything done. I've got a question. So far, everything we've done influences intensity.
Yeah.
So would you then, if you've got a sample and you want to analyze the intensity, and you need to go through, you need to apply the filters and all those things in Deconvol, both technical and biological repeats, would you then individually apply different parameters to each image? Or would you identify, for instance, if you do like add the Gaussian thing to it, the gamma, I can't remember what you call it, but that parameter there, would you get one consistent value for all the technical and biological repeats, or would you adjust that now for each individual one to make them comparable?
Yeah, so the preferred thing is to keep things consistent. So you would usually go between your different biological repeats, find some representative samples, play around with all of these parameters, figure out what is one set of parameters that works well across all of the repeats, and then apply that consistently. Obviously, that wouldn't apply to a different set of experiments, but for your set where you try to keep everything consistent, the analysis should also be...
And you would do that for across biological repeats as well?
Yeah.
Not just one set of biological, but technically
yeah, so
you would adjust, and then the next biological technical.
That's also somewhat of a tricky question, because you have to determine how much changed between the biological repeats. Sometimes it's like, okay, accidentally put a little bit more stain in your eye, accidentally, I don't know, the cells are more dense or whatever. Like, it's not always an exact one-to-one thing. Then you might want to use different parameters to get things more consistent before the analysis. But if you kept everything consistent, then the analysis can also be. Yes.
Okay.
So would you do the deconvolution everything before we do what we did yesterday, basically?
Yes. Yes, exactly. Yeah. So deconvolution, very first operation you do before anything else. But yeah.
Reduce noise and then deconvolute, or would that not?
Yeah, so there are probably pros and cons to both sides. My point more, why I prefer to do deconvolution first, is if you start to remove noise, you're removing inherently some of the modeled interferences or distortion. This pixel's intensity ended up there. You're removing that as well. So then when you deconvolve, you're removing more than you actually wanted to. So I prefer to first remove what I know should be removed, and then the noise after that. But yeah, it would also just be good because, like I said, some microscopes, you click the acquire button and then it already deconvolves the samples for you. So just make sure that if it has done that, don't deconvolve them again, because then you're going to start destroying the actual information. Okay, does everybody see that? Okay. I just saw the guy back there. So there's something called ImageJ, and then there's something else called Fiji. The Fiji is an abbreviation that stands for Fiji is just ImageJ. But what Fiji is different is it's ImageJ, but they just already have a lot of plugins included. So it's just a lot of the things that I'm doing and showing. Some of that might not be included if you just downloaded ImageJ by itself. But if you download Fiji, then everything should be there already. But it's the same software, same plugins. Nothing is different. All right, let's just see what are we going to do. We're going to open up a sample. That sample should be here. You'll see that there are 3 files. For now, just open this .czi file. We can just drag it onto it. Okay, then you should see a window like this. I'm going to maybe just show you what I did to generate the other 2 files. Is I went on split time points, because this is a time-lapse sequence, so there are actually a lot of time points there. You can check this display metadata. That is what we actually would want in the end of the day. I'm going to take it off to show you a different approach to get to it. And then you can just click OK, and then it should start importing the image. So you'll see that it's opening a bunch of windows. All of these windows are corresponding to the different time points. So where it would have been in just one single image window, now it's lots of separate image windows. Now this is, I actually should have done something else as well. So let me just go back. You want to actually say split channels as well. So just to explain what I'm doing here, when you apply deconvolution, the deconvolution lab doesn't know what you're giving it. It doesn't really care what you're giving it. You're just giving it some image data, and then it's going to start to try to apply that point split function to that image data. Now, if you give it a time-lapse sequence, it's going to think that time-lapse sequence is just one massive image. It's going to think of it as one image, and then it's going to try to apply deconvolution to it, which obviously doesn't make any sense. Another thing is if you've got channels together, you need to separate them out first, because we want to ultimately have the analysis done per channel, so not together. And you have to have different point split functions for these different channels. Okay, so to split time points, that's the easiest way I find to split if you've got a live cell. Split channels is something you can check here, so that it already splits the channels beforehand, but you can also split channels later, as I have been showing you. So you have this option, split channels on, split channels off. I'm just going to turn it off now like I had initially, and then I'll show you how you can split it up again. Now you usually don't want to do what I'm about to show you manually for every single point in time, because you're going to have 100 images, all of them have 12 time points, you now have 1,200 images, you don't want to manually open up one deconvolve, open up the next one, manually click done deconvolve. So usually we do this with macros. Now I'll turn on the macro recorder just to show you what happens behind the scenes. Let's just say, where's the record macro? There you go. Sorry, I am now doing things a little bit out of the order that I was imagining I would. And okay, importing the image, say split time points, and yeah, let me just leave that for now. You can see by doing that, there is this. This is not the right. Oh man, sorry guys. Let me just show you the one that's more helpful for the macro. Okay, so for the macro, I usually want to not just open it up, I want to say plugins and then bioformats importer, and then select my image. And then you get this window which you've seen before, and then it opens up. Okay, if I use the bioformats importer, you'll see that this thing includes a lot of other pieces of information. Let me just open this so we can see it a little bit more clearly. Come on. There you go. So it says use the bioformats importer. There is the file name of what you want to open, and then it gives you all of the parameters. Like, okay, I want to split the time points. I want to do something else. I don't know, want to have it as an upper stack or whatever. What is the order of the stack? You get all of those things, but the important one here for me is I want to split the time points. So that's how you get that information into your macro ultimately. This is not a.
So basically that menu that you showed before when you said open.
Yes.
That's automated.
Yeah, that macro would have automated it so that I don't need to go to plugins and then bioformats importer. The problem is if you don't go to this plugins bioformats importer and you just in Fiji open up the file directly, you don't get the right macro code of all of those extra features and things that you selected. But okay, so now I've got these different time points. I'm just going to close all of them so that I just have one left for now for my example. There you go, just one left. And as you can see, there are two channels here. Both of them, for some reason, is shown as green. I'm going to, for the sake of it, just make the mitochondria, I don't know, red or something. Just maybe make it a bit more distinguishable. I know both of them are red. Okay, that's not helpful. Let's just use viridis. All right, so you can, now the one is red and the other one is viridis. I don't understand what Fiji is doing. But the structures here, the one structure is autophagosomes. This is these little tiny dot things that you see there. And then the other one is mitochondria. So these are two different channels. And they were acquired under different wavelengths of light, obviously. So I want to separate them out so that I have two separate color channels, split channels. Okay, so now I've got my two images. Now before I can deconvolve them, I first need to have a point split function. To get the point split function, you need to go to image and then show info. You would have also gotten this exact same thing if you clicked on that show metadata that I showed earlier. So then here is the information. now it's already destroyed after I did all of that. Just open the CZI file. Let's just do the show metadata this time. So here's all the information of the sample that I just acquired, or that I just put into the software. Now, as I also showed you yesterday, this is not very nice for me to work with. You can extract information directly from this. You can use macros and stuff like that to say, read from this file, find the specific word, and then extract the data that's sitting behind it. But I'll show you the more manual way, which is often the way that works the best, because different microscopes call these things by different names, and you often have to find them. In any case, manually, there isn't one silver bullet thing to get it. So what I prefer to do is I just say save as, and then we can just save it wherever. Now I've got the metadata accessible. All right, so just let me know if you have gotten to this point where you actually have the metadata available. You can look at it in Excel pretty easily.
Mine opens 50 files. Something's going on. Do you have to split the time?
Well, if you want the metadata, you don't need to. So if you want to, okay, I'm just going to go from the beginning. I think I'm going to go quickly, but just look at what I'm doing, and then you'll see a couple of things here. So if I just want the metadata, I open up the CZI file. I just can drag it onto Fiji, and then I just say view metadata. There's all the metadata that I might want, and I can save this and do with this whatever I want to. Busy with. If you don't say split time points anything, you will have a single file with all of your data together, and this might not necessarily be what you want. There's no time-lapse sequence and everything, because I need to apply the deconvolution to each image individually. So to get each image individually, the simplest way is to, in this case, select, let's say, split channels and split time points. Then it will create like 50 files. So to maybe make it a little bit easier to deal with, I'm going to not check split channels, just split time points, because it's very easy to split the channels later. So now I will have lots of windows open here, and I can close all of them until I get to this one, which is my very first time point. That's where I was earlier. But the channels are still together, and then I can go image, and then color, split channels, and then I have two separate images, still Z stacks. So these are the things that we will deconvolve just now, but you basically need to get that entire massive time-lapse sequence down to this level before you can apply deconvolution.
But you do need a Z stack.
Yeah, deconvolution doesn't make sense if it's not a Z stack, because the distortion is in Z.
But if you have to do the image, you still have distortion.
But then you would remove it with other, yeah. I'm not entirely sure how you would go about doing that, if you can just apply deconvolution as if it's just one layer. The reality is if you just have a single image, you actually have no depth information. That is all flattened. It's like a maximum intensity projection, essentially, or average intensity projection. But you can probably still get rid of some of the blurring. I'm assuming deconvolution. I've just never had a situation where I had to apply it to a single two-dimensional image, so I'm not really sure.
In general, we type Z stacks, even if I just like 3.
Yes.
But it would be interesting to know if you
It's probably, yeah, it's probably going to work. I haven't tested it out, so I can't confirm, but probably it will work. It's just, yeah, you're obviously losing all of the Z resolution, but you've in any case thrown it out already, so you don't have any hope of getting it back after deconvolution. Okay, does everybody have a CSV file open?
I just want to
Yes?
I just need to submit the data file, with that file, without saving.
Sorry, yeah, it would be best if you can save it as a CZI file. So you've got your metadata here, then you just go File, Save As, and then you just save it as a CSV file somewhere on the computer. It doesn't matter where. And then you just open that CSV file.
sorry, Vincent?
Yes.
Do you need to have 2 different files, each for each color metadata file?
No, there's only one, in this case, there's only one CZI file. Both channels were captured into this one single file, and the single file contains the information for both channels. So there's only one set of metadata for one file, one acquisition run.
If we save it as separate ones, because the first round we did, you saved 2 files, so that's still okay.
What you mean, like these two files not for the different
The one
channels?
for grade and then the other.
Yeah, so you need to have, okay, there are two things here. We need to have the, for my entire sample set, which was a single file in this case, I need to have all of the metadata for that. That I get from that one file. There's metadata associated to everything. And then I need to apply deconvolution later, but for that I need to have separate images. So,
So we need 3 CSV files.
no, one CSV file, which is the metadata, and then 2 image files, which are the 2 images that we want to deconvolve just now. Okay, so these 2 images, which I showed you how you get them, I was pretty quick, but I've given them to you in the folder as well. So these 2 exact images you're seeing in front of you, you don't need to get them necessarily. I've quickly shown you how to get them, but you can just open them up directly in Fiji.
Would it matter because I've got the Excel file, but it's not separate. It's the commas. It's all in one column, so I don't have like a.
Okay, so maybe there's a just a setting I need to comment on in Excel. If you go to File Options, okay, you'll see this window here. Okay, then under Advanced, I think. Then here where it says decimal separator, you just need to change that to a point. So it's in South Africa it's by default a comma, and then you just need to change that to a point, like a dot. And then it should automatically open it up fine. That's one approach. Another approach could be that you, let me just try to force this now. I'm going to create a new file here and take that CSV file, open it with, just take VS Code. Okay, yeah, so you see in the actual CSV file, the way it's actually stored is it uses a comma to distinguish the first column and the second column, so that comma is the column separator. So if you open it up, let me just select all of this. And then paste it in Excel. If it looks like this in your case, it's just all of it is just in one column. Then you can select that column and then say data and then text to columns. It's going to show you for a second, data, text to columns. And then if you click on that, you say it's delimited and it's delimited with a comma. You only want to select the comma thing there. And then it should separate them out so that you now have them as separate columns. But I personally work with enough CSV files that I'm frustrated by the default settings in Excel that I just go and change the default settings. Okay, but everybody got to this point. You've got your metadata open in Excel.
Yes.
I'm glad, thank you. All right, so now we need to generate the point spread function, and we first need to know what do we actually need. I was mentioning some of this very quickly in the presentation, but let's see that point spread function generator that we installed earlier. It's plugins PSF generator. Just open this thing up. Okay, you should see this window here. All right. And now you'll see that those names that I mentioned earlier, these are the different ways in which you can generate the point spread function. They all require slightly different information. They kind of tell you in which context it's appropriate, but this is as clear as mud. It's not telling you anything really, is this appropriate for you or not? So that's why we just tested them all out to figure out what actually works for our microscope. In our case, the Bourne Wolf one was the best. So for this data, this image, you should please use the Bourne Wolf. Now if you look at the parameters that they're asking us, they ask for a refractive index, immersion. Now you should be able to just know this if you actually acquired the samples. If I acquired this with oil, it will always be 1.518. That's a fixed reality. But you can also find this in your metadata. I'm going to try to keep both open here so we can work back and forth between them. If I was to just say find, which I just press Control F, and then you can search for refractive index. And then you should find a row somewhere along all of these hundreds of lines of text, which is called refractive index. Let's make it a little bit larger. Just going to highlight this one for now. There you go. Maybe it would also be good, the ones that are useful, I'm copying them over to a second sheet because I want to see them all together just now. Okay, now let's just look at what the other metadata information is. This size C is the number of channels. Well, as we saw, we had 2 channels. Size T is the number of time points that I had in my time-lapse sequence in my movie. I'm not really using that information right now. I'm working with a single slice. So what I care about, according to this thing, is I need the size of X, Y, and Z. I need the resolution, essentially, of X, Y, and Z. That's this bottom row here. So here I get it. The size of X, Y, and Z. So I'm going to copy these values over as well because I will use them just now. Okay, so what else do I need? This bottom part is not sorted. That top part is sorted. I need the wavelength. And remember, this is the emission wavelength. So I'm going to search wavelength. Come on, wavelength. Okay, now you'll see different values here. This is a little bit trying to read between the lines thing sometimes. But there's a laser wavelength. Now, if I see laser wavelength, I think excitation wavelength, not emission wavelength. So I'm going to skip over that. If I keep going on, it's more wavelength things. Yeah, these might be actually values to use. I'm just going to show you a little bit further. There's another value that's a bit more interesting. Just want to make this wider. Sorry? What did you say? Yeah, I just want to show you, yeah. So yeah, the detector wavelength range. This is the more interesting one because this is the range over which it was actually capturing. Now, in your case, hopefully it didn't show this notation. Let's see if I can. Okay, that's a very small number. I'm just going to multiply this with 1 million. No, that's 100,000. That's 1 million. I think I can maybe even multiply it with a billion. Yeah, so now I'm getting it to nanometers. So this is now the unit in nanometers. Come on. Now, as I'm saying, this is probably the more helpful one to look at because if you want to decide where is my wavelength that I'm going to choose, you should typically consider what was the range in which it was emitted. So I'm going to, yeah, there it tells me now which one is 1 and 2. Oh man, this is going off the screen. Okay. So I want to match the 1 and the 1 and then the 2 and the 2 in this case. So I'm going to say for, let's just call this one channel 1. It will be this one minus that, not minus that one, average, oh, come on, average of that one and that one, and then channel 2 will be the average of the other 2. Okay, so these are the 2 emission wavelengths that I'll be using. You could now be a little bit, like I say, more rigorous with this and decide what was the actual intensity in it, but that would be a good one to use. It would also just be good to quickly scan through what else do I see here in wavelength, because the wavelength is ultimately the core parameter that makes sense. Here is basically that same information represented just in ranges. You can also use this. Let's just see what else I get. There's an emission wavelength. The irritating thing with this one is there's only one emission wavelength, which is 5 to 3. So what do I do with that 5 to 3? Like which one is it now? Is it now the channel 1, channel 2? I'm not giving that information. So I'm going to just use what I found before, where you see 5 to 3 is in any case what I calculated. So that seems like a reasonable assumption here. So I'm going to copy this information across. Based values. So this is the wavelength.
So we've got 2 numbers of wavelength on the Excel spreadsheet.
Yeah.
For the DSF generator, it's only one option.
Yes. Okay. So as I briefly mentioned earlier, you need to have a different point spread function for different wavelengths. because the amount of distortion depends on the wavelength. So we will be generating two separate point spread functions and then do the deconvolution separately for the two different channels. Okay, let's see what else we need. We need the NA, which stands for numerical aperture. So I'm just going to search. Sometimes it might be called numerical aperture and you need to find it in that way. In this case, I just know that it is actually called lens NA. And in our case, it is 1.46. So I can copy this one over. Okay, let's look what else we need. We need the pixel size XY, which should, the reason why I say pixel size XY is because X and Y is going to be in a perfect grid. So the width of an X direction for a pixel is going to be the same as the height for a Y pixel. So that's why it's the XY pixel size. And this is how much nanometers is represented with a single pixel. That's on X. And then how much nanometers is represented with a single step in Z. Okay, so let's just search for those things. Just thinking this is actually the values, but they are not that easy to interpret. Let me just see if I find something else. Here it is. Yeah, here's another set. It's the same values. That might be a little bit more clear. So the scaling, this is now the acquisition mode. The scaling in Z, the scaling in Y, scaling in X tells you how many nanometers is represented by that structure. Again, this is in the units of meters for whatever reason. So I'm just going to time this by, again, a billion. That's 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay. And just format it like this. Okay, so in X and Y, my scaling, as I would expect, is 83 nanometers, and in Z it is 350. And I think I have everything I need now. Okay, so now I can start populating things. Refractive index is 1.518, say 18. My, let's just make the accuracy, everything is slightly inaccurate, so you can now choose how much inaccuracy you want to tolerate. The better you choose, the longer it's going to take. I'm just going to choose better. The wavelength, let's just start with channel 1. So for the channel 1, I'm going to use 523. Numerical aperture is 1.46. The pixel size XY is 83. And the Z step is 350. The resolution for X is 1024. For Y, it's also 1024. And then the Z step, or the number of Z slices is 6. Okay, so once you've got all of that filled in, you can click Run. It's going to take a little bit of time. And then you get an image that pops up. So this image is your point spread function. You can now go through it in Z, and you'll see that there is actually some variation in Z. It should be symmetrical as well. Okay, now I would like to save this because I don't want to go through that entire hassle of generating again. So I am just going to say I've now selected my point spread function, then I go File, Save As, a TIFF file. Okay, and then I will just call it whatever it is, but just make it very clear this is for channel 1. This is the one I should use for channel 1. Okay, now I'm going to repeat this entire operation, but luckily the only thing I need to change now is the wavelength. So this is 698, or just try to see if I can add some decimal point there. And this is, everything stays the same, it's just the wavelength of light that has changed now for channel 2. I click Run again. And there is now the point spread function for channel 2. It should look slightly different. If you were to compare the two, you should notice that they're not identical, but the wavelengths are in a similar range, so that's not too far apart. Again, File, Save As, TIFF, and I'm going to just make clear that this is now channel 2, and save. All right, so just to start from a somewhat clean, do you want to save changes? No. somewhat clean slate. I'm just going to close everything here. Yeah, you would just do one channel at a time. So you would, well, you could technically do them in parallel, but it will be a macro, as Elisa was saying. So this point split function generator, you can't really do with a macro. I haven't found a way to call this point PSF generator from a macro rather. So I can extract all of these values if I know what the names are here. You can write a macro that would automatically, you just open your image, click extract values, and boom, all of the values pop out directly at you. You don't need to scroll through the thing and figure it out. This, getting the actual point split function generator, you have to do manually. But then once you have your PSFs, then the hard work is kind of done. Then you just offload it to a macro that then, and I'll show you how to record the macro. That just simply reads one file at a time. It automatically splits it according to time points, according to channels, and then it just applies the correct PSF to each of these files, saves it in a new location, and yeah, there you're done. So if you scratch around on my GitHub repository, there's this FMPP. Why I called it that, I can't remember now. Fluorescence microscopy processing pipeline, apparently. In this thing, there is something get metadata for PSF. So this is a macro that basically just you give it your image, and then it looks for this string. And then wherever it finds that string, it then assigns it to a value, and then at the end of the day, it just prints out all of your values out. As I was just saying, this is different for every microscope or whatever you use. So this is a little bit of a pain, and this doesn't necessarily generalize, but if you figure it out for your microscope, your image data, it's got this format, it's got this labeling, then you can extract it automatically with the macro. I'll show the macro just now because I don't want to open it up later, but there's also one for deconvolution. Okay, so the general process that I usually take is I have the PSF file somewhere on my computer. It's a fixed file for my data set, and then I have a folder which contains all of my files. And then I just set up some paths for where's my raw images and where do I want to save it, where is my output images. Essentially, just create directories if I need to. I know I'm going through this quickly, it's not the point that you learn and figure out what's going on here. Then I list the files in my directory. So I then have a list of all of the files I want to deconvolve. Go through a series of things here. Here's that bioformats that I was mentioning earlier. I just say open that file that I just put together. In this case, I also say split the time points. And then I call the number of images that then open up the number of frames. This is the number of time frames, essentially. And then one frame at a time, this was only a single channel image, so I could just do it like this. I then just run that deconvolution lab, which we will run just now. And then I say, open up this file, use this PSF, but all of these things are generated for you. Use this method, use this number of iterations. This entire line, you'll see just now, will be generated for us. And then I just run it on all of the images, and at the end I just combine it back into one big time-lapse sequence again. So this is giving you a bit of just a feel for it. Macros allow you to just click run, and then everything does it by itself. You can use this as a base, ask ChatGPT how to change it for your purposes. It is available online. But let's now just, everybody's got the images, everybody's ready, let's do the deconvolution. Okay, so what we do is you don't technically need to open it up, but I want to show you if you were to have it open, I'm going to choose the one that says C1, that's channel 1, it starts with C1, opening up that image, and then I take the C1 PSF, I open up that image, so now I've got these two images open. And don't let the colors fool you, this is just a lookup table. It's not that there is actual color here, it's just a lookup table which is rendered in that way. Now I can run my deconvolution lab. So I go for plugins, deconvolution lab 2, and then deconvolution, or maybe I must just turn on my macro recorder because I want to show you that later. Okay, there's the macro recorder, plugins, deconvolution lab 2, and then the first one. No, you don't need to do the macro. I just want to show you that that code can be generated. Okay, so if you select that, you get this little window that pops up. Now you choose which is the image that you want to deconvolve. Now you can either get an image directly from the computer, so you don't even have to have it open in Fiji. You can read it directly from the computer, so that is very helpful. That's what I was doing in the macro. But if it is open in your, actually in the macro I was first opening it up, but you can read it from the computer directly, it doesn't matter. If it's already open in Fiji, you can say, select this option, get platform image from the platform. Then it's going to ask you which image, because there are 2 images currently open. So I choose the image file, click OK. And then the next one is the PSF. Where's the PSF? Again, image from the platform, because it is currently open. And then I just choose the PSF, click OK. And you'll see that there is actually some code that's generated at the bottom there. It's tiny on my screen here, but there is a little piece of code there that that is the code that you can put into the macro. So that is macro code already, but we'll see it's going to generate some, or it should generate a macro on the side just now. Oh no, actually, I'll try to demonstrate how the macro code can be generated. Now I need to choose an algorithm, and as I was trying to make a point earlier, the algorithm that we found to work the best is Richardson-Lucy total variation. It could just be Richardson-Lucy as well. The total variation in our case worked a little bit better. So you choose that one. Then you need to play around with these 2 parameters. How many iterations do you want to run this thing? There's always a trade-off. You need to play around with this. If you choose 5 iterations, it might be sufficient. It might not be sufficient. If you choose 10, it might be you're destroying your data. It might not be the final point. You have to play around with that, try some different numbers of iterations for some representative samples and decide, this is a good value, I'm going to apply this to all of my data. So I'm just going to choose 10. And then the regularization is, it's also a parameter to play around with. This is trying to keep structures basically. So how much are you going to give it help? Again, play around with this. I've usually used something in the order of just this value, 10 times, or 1 times 10 to the power of minus 4. But yeah, you can again play around with that. Okay, and then finally I can click run, and then it will start the entire deconvolution process. If everything worked, you should have an image open up after the entire process finished. In this case, it didn't take that long. One image can easily take minutes to hours, depending on how large it is. Yeah, so let's just quickly compare what we're looking at here. If I just take my lookup table, let's just make this Viridis. This one also the same. And let's just, maybe I'm just going to show you a quick trick. If you go to Analyze, Tools, and then Synchronize Windows. I'm going to show you Analyze, Tools, Synchronize Windows. Then, and just say Synchronize All. Then, if you change the slices in one, it changes it in all of the images to be the same. And then also, if I hover with my cursor over a certain place, it will show where that cursor is, where the position is in the other image. I just need to enhance this a little bit. Okay, that one's better. And then this one, this is too harsh. Yeah, so you need to now see that actually the points that I'm interested in are these bright dots. That's the actual autophagosomes. So I can bring everything low. I can bring the noise up if I want to, but actually I want to make sure that the structures I'm interested in are not saturated. So this is a kind of a good place for that. And then here, a similar approach. Make sure that it's not that saturated. So hopefully you can see that a lot of this blurriness, fuzziness that was there if you're on this screen, but if you maybe just, I'm going to exaggerate this to make it a bit more clear. But a lot of the fuzziness is actually now gone. And that's exactly what we were hoping to see with this deconvolution. All right, so let's just do the other one to wrap this part up. I'm going to see if I can get it to do the macro thing properly now. So I'm going to go to Deconvolution Lab, and then I'm going to click Run this time. And now select, wait, I must first open up my images. So it's the second channel 2, and then this one. Okay, so there's my channel 2 PSF and the mitochondria. Plugins, Deconvolution Lab, run. And selecting from the platform channel 2. And selecting from the platform point spread function, Richardson-Lucy total variation, 10 iterations, that number of things, and then run. Okay, again, the backrow didn't create properly. So it seems like the way that they want you to set it up is actually to copy that line of code, as I showed you before in my example. Where is, I think I had it here. So that you actually have that image platform PSF file all specified, and then the algorithm also specified, etc. But they just give you the code. They don't actually generate the macro properly. Okay, so there's my output image. I'm going to do the same, make this viridis, and in the input image, also viridis. Analyze, tools, synchronize windows, synchronize all. Okay, another thing is, usually the zoom also synchronizes, but anyways, didn't quite do that now. No, but if I drag the one around, the other one also drags. So very, very helpful to have synchronized windows. But yeah, you should be able to see that the structures are a lot more defined now.
Yeah.
Because previously, what we were looking at here is not the intensity on a single Z plane. It's actually some of the intensities from the lower planes bleeding through. And if I now scroll through this, you can see that, let's say, for example, this structure there, here I can see some intensity, but that's actually a lower level, a lower Z level, which the intensity is bleeding through. And if I was to just scroll through it, I will find there's the structure. That is actually where it's supposed to be. So I've got everything, especially in Z now, very nicely in the place where it's supposed to be. And in X, Y, everything is just sharper. Yeah. Okay, any questions about deconvolution? Yeah.
Is it really helpful if you don't have a clear image, or is it still?
Yes, so if the point is that this intensity image is not a true representation of what I have. So if I was to start to calculate the average size of something, it would say that in Z, the size extends all the way to this level, where in reality it didn't. So I would have wrong sizes or wrong volumes or areas associated to structures if I don't deconvolve them. But if you work with big structures, like a full nucleus cell is filling up the entire frame, it's going to have a minor impact. With mitochondria, where it's these filamentous strands, there it has much more of an impact. Okay, I think that's it on deconvolution. And oh yes, I maybe just want to make this comment. This is just a helpful picture, maybe to have as a reference stuck on somewhere, because when we work with these values, we lose a little bit of track of what color is actually associated to it. And then you might say, hey, okay, my I don't know, my GFP, which was clearly in the green range, I'm going to put in a wavelength here of 650. It's like, well, that doesn't really make any sense. There's no way that the wavelength could have shifted that far down from excitation to emission. I'm expecting it to be in this range. So it should be somewhere in the 500 range, and we found out 523, which makes a lot of sense. So just having that intuition, how does the wavelength match to the color? Because there is color that you observed, just to make sure that you have, it's just a sanity check, just a good thing to keep in mind. Yeah, here's the instructions for what we just did. Here are some of the results that we just saw.